Oak Ridge. We have a high flux isotope reactor where the Biosands is located. That's a continuous reactor that produces neutrons by fission of uranium. And we also have a spatial neutron source, which is an accelerator-based neutron source. And that produces neutrons by bombarding a mercury target with a proton beam. This is a schematic of the layout of the instruments at both of these facilities. I've highlighted about 11 instruments here, and they're the instruments that we primarily use for biological research. So in total, there are 30 instruments at both facilities. So the Biosands instrument, as I mentioned, is located at the high flux isotope reactor. This is a schematic of the cold guide hole, and Biosands is circled here. We will have a virtual tour that Luke is going to give us later on of Biosands. And then the biodeuration lab is located at the spatial neutron source. And then this picture shows the detector tank for the Biosands instrument. And you'll see a lot of this later on in the virtual tour. So just a quick note on how we're organized. This is our team. Volker, Venke, and Wellington run the Biosands instrument. Luke is our scientific associate that supports Biosands. And then Kevin, Shu, and Hong Hai are part of our biodeuration effort at the spatial neutron source. So this is an overview of Biosands. You can see a schematic in the instrument here. So I'm pointing to the collimation system. This is essentially the neutron delivery system to the sample area where we have different sample environments. And then this is the detector vessel for Biosands. So these are some of the key features of Biosands. We have a dual detector configuration shown down here in the pictures below. So we actually have two detectors in the detector vessel. This is the wing detector shown up front here, and then the main detector is seen at the back. So this gives us a very large dynamic Q range or spatial range that we can access anywhere from about 100 to several hundred nanometers in spatial range. We can perform simultaneous scattering and transmission measurements. That greatly speeds up our measurement time. And as you'll learn later, we've got a very versatile sample area for doing a wide variety of different types of measurements. So we're continually upgrading and developing our instrument. In the past few years, we've completely replaced and upgraded our collimation system. We've upgraded our data acquisition system and also our data reduction. And we've also developed some new types of sample environments that you'll hear about, such as a robotic sample changer. And we're in the process of actually installing another detector in our detector vessel, a mid-range detector that will help to increase our Q coverage, or the spatial range that we can cover by the instrument. So our biodeuration program is a critical part of our program because it allows us to produce novel types of biomolecules that we can then perform different types of experiments with that you couldn't do with other techniques. So we can characterize higher order macromolecular complexes and see individual components of these complexes. We not only support the Biosands program, but we also support a lot of different types of experiments across the suite of instruments at the high flux isotope reactor and the spatial neutron source. So we also support neutron crystallography, neutron dynamics or inelastic neutron scattering, as well as we also, in addition to producing proteins and nucleic acids and lipids, we also produce different types of deuterated biomaterials such as cellulose and also recently chitosan. So This allows us to do a large variety of experiments. So we have a very well-equipped laboratories to support this program. Kevin will give you a virtual tour of our laboratories, and you'll be able to see some of the equipment we have. One of our specialties is fed batch fermentation, so we can do large high density cultures of both E. coli and yeast to produce deuterated molecules. We also do shake flask for both bacterial and eukaryotic expression studies. So we routinely now work with insect cells in the labs, and we purify a wide variety of proteins, both soluble and membrane proteins. We have equipment to purify and characterize different types of biomolecules. So we have various ACTA systems for purifying proteins. And we also have a lot of different types of spectroscopies, such as CD, circular dichroism, and UV visible spectroscopy and fluorescent spectroscopies, as well as access to GC mass spec for characterizing lipids. And then we also support different types of biophysical techniques, such as small angle X-ray scattering and light scattering, as you'll hear about later. So just a quick overview of our user program. There are different ways to get access to our facilities. The primary way is through the general user program. We allocate 75% of available beam time through our general user program. These are typically for two to 3 days experiments, and we have 2 proposal calls coming each year. And our next call is coming up later this week. This month. If you need biodeuteration, then you would request biodeuteration as part of your GU program call. So we also allow proof of principle experiments. So if you have an idea for an experiment that you're not sure if it will be successful, you can apply for 1 day of beam time just to do some test measurements to see if your experiment would be feasible. These are actually internally reviewed, and we can schedule these quite rapidly once they are approved. Since the COVID-19 pandemic, we also, in some cases, can offer remote access. And remote access can be applied for both through the general user and the proof of principle mechanism. Using this approach, the user then will send us the samples, but the user then can control and run the experiment themselves. So we keep back about 25% of the available beam time for program development activities or in-house research. And I'd also like to bring your, I don't know what happened there. I'd also like to bring your attention to 2 other programs we have. We also have collaborative access programs, one of which is with the National Synchrotron Light Source that's located at Brookhaven National Lab. So you can get joint access to the LIX beamline, which is small angle X-ray scattering beamline, and BioSAMS through one proposal. That allows you to both get X-ray and neutron data for your experiment. And that is generally applied through our general user program. More recently, we've got involved in another DOE program that's run by the Biological Environmental Research Office, and that's called Facilities Integrating Collaborations for User Science. So the FICUS is the acronym for that. That actually gives you access to the Joint Genome Institute at Berkeley National Lab, the Environmental Molecular Sciences Laboratory at Pacific Northwest National Lab, and BioSANS as just one proposal. That started in last year. If you want to hear more about that, please contact us and we can give you an idea of the types of projects that are funded through that system. So that's the end of my talk. I've just listed a few resources here that we can make available to you. You can find a lot of information at our website, neutrons@ornl.gov. Once you become a user of our facility, you automatically get signed up to the SNS HIFER user group, and you'll be on their email list. The CSMB has its own website that you can access here. And then I would also draw attention to the Department of Energy BEO Structural Biology portal. This is a website that highlights all the different types of biological research that you can do at both the synchrotron and neutron sources across the US. So as I mentioned, we have a proposal call coming up. The deadline is September the 21st. So if any of you are interested in submitting a proposal, please reach out and contact one of the team and we can help you submit that proposal. Okay. Thank you very much. So are there any questions right now that you guys have? We've got a couple of minutes for questions. No questions? Okay.
One comment, maybe?
Yeah.
We are recording the presentations, is my understanding, so we should let people know about that. You normally get a little kind of warning in the Teams thingy anyway.
I don't think we have started recording them. Oh, you have?
I pressed the button.
Oh, you did? Okay, brilliant. Okay. So I hope that's okay with everyone. Let us know if you have any concerns about that. I should have mentioned that as part of my introductions.
Thomas has a question.
Yeah. Thomas?
Hi. So maybe I missed it, but I know that a lot of other beamlines have to have a significant period of downtime for upgrades and these types of things. Do you have a timeline for future upgrades that will make long-term downtimes at the Biosands beamline? Is that planned? I assume that there will be upgrades planned in the future.
Yeah, the next major outage we will have will be in 2028 for the beryllium reflector changeout. So up to that point, we expect to be running 5 to 6 cycles per year. And I don't know if the month or the day in 2028 has been decided yet, but we've got a few more years before we'll have a major outage.
We will be installing the mid-range detector that will probably cause us a little bit more of an outage than usual, but I'm talking about maybe a couple of months rather than a year or something like that.
Yeah, hopefully one cycle to commission it, but no more than that. Yeah,
that's correct. Yeah.
And I will mention that the spatial neutron source is going to have a major upgrade in when does that start? Next year, right? Late next year. So that would put
like February.
Yeah. Okay. So that will put the EQ sands out of operation for about a year.
So the really long outage, the PPU proton power upgrade outage, the really long one, starts late in next year, so like in August or September, and then runs for quite some time. Yeah. But that's, like you said, that's the SNS. That's the other instrument. Yeah.
So you should expect to have access to neutrons here for the next few years. But thank you for bringing that up. I think it's important.
Thank you.